The overall goal of the following experiment is to measure kinetics of protein-protein interactions and the allosteric effects of small ligands on those interactions using Bio-layer Interferometry (BLI). This is achieved by preparing one of the proteins with specific biotinylation and immobilizing it on the surface of streptavidin-coated biosensors in parallel. As a second step, the sensor-bound protein is exposed to buffer containing its binding partner, and binding is measured in real time by its effect on optical interference at the sensor surface. Next, each sensor is transferred to buffer without the binding partner in order to measure real-time dissociation of the protein-protein complexes. Results obtained allow determination of the kinetic rate constants for binding and dissociation of the interacting proteins and thus their binding affinity. Further results show that BLI can detect the effects of small ligands on the rate of dissociation of the protein-protein complex.
The main advantage of this technique over existing methods such as surface plasmon resonance is that BLI is not overly sensitive to refractive index changes, so the binding and dissociation steps can include changes in small ligands to test their effects on the protein-protein interactions in real time.
Though this method can provide insights into kinetics of protein-protein interaction, it can also be applied to different combinations such as protein binding to immobilized DNA or RNA, or liposome binding to an immobilized protein.